Okay, we're just reviewing the 2021's paper, right? Not writing any answers, just discussing them. I would prefer if everyone writes their own answers because that will benefit you more. But I'll give some input. So the first question in Module 1 Bioenergetics says here, an experiment to observe the interconversations of compounds found in the nitrogen cycle was carried out by a student. 150 centimeter cubes of a solution of urea was mixed with 50 grams of well aerated loam soil. The ammonium, nitrite and nitrate concentrations were immediately determined semi-quantitatively by using test strips that produce color changes. The strips were then compared to the specific color scale, which gave a measurement of the ion in parts per million. The mixture was sampled for both ions and 6 hour well at well in it says here 6 hour intervals for 24 hours. So figure 1 is a graph which represents the changes of the ammonium and the nitrite ions in the mixture over the 24-hour period. Right? So the triangles would be the nitrates and the rhombuses would be ammonium, I guess. Right, so that is a change in concentration there. Okay, so let's look at this. Alright, so here we're supposed to construct a table to represent the data in figure 1. Right, so let me just check something else. Okay, so we're supposed to construct a table to show this. Now, can somebody explain to me, like walk me through how you would construct that table? So how would we construct this table? No? Okay, everyone knows how to construct the table from this?
Could you split it in three and put the period of incubation, and the other two sections would be labeled the concentration of ammonium ions and concentration of nitric acid, and you put the values to match with it?
Okay, so this would be, let's say, column 1, right? This would be column 2, and this would be column 3, and then you just put in the values.
Yeah.
Alright. What would be the title of the table?
Table showing period of incubation, along with the concentration of ammonium ions and nitrate ions in ppm.
Okay. Anyone else has another suggested title? Some persons tend to just use table showing and then put back production of ammonium and nitrate ions over 24-hour incubation period in loam soil with a solution of urea. Some persons tend to like just use what the figure title is and just put table showing in front of it. Do you guys think that would work?
Yeah.
Alright. So the first question is really just a table. Does it assess biology knowledge? No, you just have to really know how to draw a table and it should be fine. 5 marks, right? It's really a steal, to be very honest. Here now, a diagram of a simplified nitrogen cycle involving plants in a natural ecosystem is shown. So now we have to label all the processes shown. So what would be A? So from living plants, organic nitrogen in the soil, what would be A?
Would it be nitrogen fixation?
Hmm.
Would it be decomposition?
Okay. Hold on. Alright, so living plants and organic nitrogen soil. So we're saying decomposition or nitrogen fixation. Define nitrogen fixation, anybody? What is nitrogen fixation in your own words? Michaela, Rodella? No one? Do we completely understand the nitrogen cycle? We didn't know nitrogen fixation, nitrification, denitrification, ammonification. Okay, so nitrogen fixation involves the nitrogen within the air being converted to organic nitrogen, and there can be biological nitrogen fixation, right? And this involves a symbiotic relationship between rhizobium, right, and some amount of rhizobium, right, within the root nodules of nitrogenous plants, or leguminous plants. Sorry about that. So yeah, so it would be nitrogen fixation for E, but A would be decomposition or death. So there would be decomposition, death, decay, whatever you want to write. So it's gonna be decomposition or decay. It's gonna give us nitrogen within the soil. And this means that the proteins, right, within the plants, and anything that really contains nitrogen within the plant, right, which would most likely be proteins, right, would be left in the soil. And then there's going to be a deamination process, right, as well as ammonification by ammonifying bacteria. So we're going to see stuff like Pseudomonas and Clostridium, right, producing ammonium ions within solution. And this will be ammonification for B, right? For ammonium to nitrates, right, this would be nitrification, right? So we're gonna see things like nitrosomonas and nitrobacter being involved within these processes. So nitrosomonas, I think, brings ammonium to nitrites, and then from nitrites to nitrates would be azobacter, right? So we're gonna have those. So that would create the nitrates, and when nitrates go back to the air, we're gonna have denitrification, right? So the the nitrates return to the air, right? So we're gonna have the reduction of the nitrates. That would be denitrification, right? So I made small explanations of the entire nitrogen cycle, right, and put it on the channel, right? So all of it should be there. So these dotted lines here, what do you think these dotted lines would be? Those dotted lines from nitrates to the living plants and from ammonium ions to living plants, what do we think they will be? Anyone? What do we think they would be? Okay, so that would be assimilation or absorption, right? So animals may also be involved in the nitrogen cycle. Extend the diagram in figure 2 to illustrate the involvement of animals in the nitrogen cycle. How would animals be involved in the nitrogen cycle? You're gonna have animals, right? Living plants, and animals will also decay, right? So we're gonna have that there. That's how they're going to be re-implemented into the cycle. I'm not hearing anybody try to answer the stuff. I'm not sure why, but I'm just going to move on. Energy is needed to metabolize nitrogen compounds within organisms. Under anaerobic conditions, the respiratory pathway differs depending on the organism. State two similarities between fermentation reactions in animals and yeast. So we should know how fermentation works in animals and how fermentation works in yeast. So it's lactic acid fermentation within animals and ethanol fermentation within yeast. What are two similarities between these fermentations? All right. So there are some similarities between them. So the two fermentation processes that you'll see, right? Both of them take pyruvate, right? So both the initial the reactant that they both take is pyruvate. So both take initially pyruvate, right? And both would produce 2 ATP, well, ATP in one turn of its cycle. So both produce ATP. Those could be similarities. Those could be two similarities between them. So both takes pyruvate and both produce ATP. Both are anaerobic already, so that's really said about the question. So I don't think that's a problem really. So we have that. Explain the function of fermentation reactions within organisms. Could somebody explain that? What is the function of that? Of fermentation within organisms? Okay. So fermentation, we would remember the steps in respiration, right? You're gonna have glycolysis, then you're gonna have your link reaction, then you're gonna have Krebs cycle, then you're gonna have your electron transport chain. Now, in order for Krebs cycle to go right and lead into the ETC, we're gonna need oxygen there, right? So if we don't have oxygen, if we're working in anaerobic conditions, generally fermentation would be like a backup, right? So a backup generator, in a sense. So it's a backup reaction for those organisms that can respire aerobically, and it is the main function or the main mechanism through which organisms that function anaerobically would produce energy. So fermentation is that process, right, that allows organisms to still produce some amount of energy for general metabolic function, right, while with the while in the without oxygen, right, without the presence of oxygen. Within organisms that do respire with the oxygen, fermentation allows it to even produce some amount of power as a backup while cells are starved of oxygen, right? But within tiny organisms that like don't respire aerobically at all, it would be the primary mechanism of ATP production, right? So you could explain the function of fermentation reactions in organisms that way. All right. This now, let me have a look at something real quick. This is real cool. Would that be all of module 1? Yes, it would be. Okay. So let's have a quick look at this, right? So It's a bit blurry, isn't it? Okay. The process of oxidative phosphorylation is very important for the supply of respiratory energy under aerobic conditions. Discuss the importance of the following components of oxidative phosphorylation. What is the importance of the proton gradient in oxidative phosphorylation? What is so important about the proton gradient in oxidative phosphorylation? We could remember that oxidative phosphorylation, right, would happen within the mitochondria, and we're talking about the inner membrane of the mitochondria. So this is talking about the ETC, well, the importance of the proton gradient to the ETC, right? So we know that as we go through the ETC, we're going to have other types of complexes that pumps hydrogen, right? So we're gonna have hydrogen pumps, right? Making that clear. So usually when mechanisms pump hydrogen across a membrane, right, it usually creates a type of chemical energy, like bioelectricity in a type of sense. It charges the intermembrane space of the mitochondria, right? So it would be creating such immense charge on one side. Now we're gonna have chemiosmosis happening. So as the hydrogens create this concentration gradient or this proton gradient, right, it's going to create a differential charge across the inner membrane, right? So when these protons start to move through the proton gradient, it's going to result in chemiosmotic reactions in ATP synthase, which produces ATP from ADP. So one of the main functions, right, of the proton gradient would be establishing, right, charge differences and allowing for the protons to, what you'd say now, initiate chemiosmosis, right, for ATP synthase to produce ATP. And that's one of the major processes or major functions of oxidative phosphorylation, right? So if I could explain in a different way, right? Well, simply putting, the proton gradient is produced, right, by pumping protons, right, during the electron transport chain. Now the protons flow down their concentration gradient into the matrix through the membrane protein ATP synthase, if we could remember, and that causes it to spin and catalyze the conversion of ADP to ATP. So that would be that. The synthesis of ATP. So we know the importance of ATP to be generally the basic unit of energy within the cell, right? So we know that ATP, right, has, well, which is adenosine triphosphate, it has 3 phosphate groups on it, right? And a lot of energy is stored within within these phosphate groups. So the synthesis of ATP, the importance of it is that it creates energy not only for not only for it to be readily used, but for some amount of energy storage, as energy is stored within these molecules. So we know that when we break the bonds, right, on ATP to create ADP, it releases a jolt of energy, and that is important for for most metabolic reactions, right, that require energy. So active transport mechanisms, some catabolism, right, some anabolism, something like that, right? So some of these metabolic processes would be used, right, or would use energy, right? So as Neil said, yes, used for energy for the processes in the cell, that would be really important, right? ATP and the use of oxygen. So we know that oxygen is really, really, really important. And we know that without oxygen, the ETC will not be reached, much less, the Krebs cycle will not be attained, right? Much less the ETC. So note that oxygen acts as the final electron acceptor. So while electrons are moved through the ETC from complex 1, so we know that at complex 1, NADH releases electrons there, and at complex 2, FADH2 releases electrons there. And these electrons will move through oxidation centers, right, creating the proton gradient. Now, after the proton gradient is established, as we talked about before, protons will move down the gradient to produce ATP. But notice that these electrons are still there. These electrons need to be accepted into oxygen, right? And then the oxygen would be reduced to produce water, right? If oxygen is not there as the final electron acceptor, electrons will be allowed to be loose within the electron transport chain, and this can create free radical systems. And as we note from chemistry unit 2, free radicals are usually unstable elements or unstable substances, right, that have an unpaired electron. So that will then create some havoc within the cell, right? So it can destroy protein, it can destroy genetic material, right? So we don't want a lot of free radicals within the body. So this is why oxygen is here as a final electron transporter, right? So it makes sure to take up these electrons to prevent the creation of free radicals, right, and allow for the body to remain stable. And I know that a lot of persons would have done Cape foods as well, so this is importance of, this is why we consume antioxidants and stuff like that to prevent free radicals from entering, from being formed within the body to destroy protein mechanisms, right? So this is why oxygen is here. That's the role of oxygen as the final electron transporter, right? And that's really it for the module 1 of 2021, right? And it wasn't so difficult.